Chapter 12, Segment 3. Dysfunctional androgen receptors can block male masculinization. Androgens are important for masculine sexual differentiation. Androgen insensitivity syndrome is caused by a mutation of the androgen receptor gene that prevents tissue from responding to androgens like testosterone. The Wolffian dogs can't sense testosterone, so they regress. The external genital tissue forms labia and a clitoris. At puberty, women with AIS develop breasts. This depends on the ratio of estrogenic to androgenic stimulations. Since androgen-sensitive individuals receive a small amount of androgenic stimulation, the estrogen to androgen ratio is high. However, because of the lack of ovaries or a uterus, they cannot menstruate and are infertile. In all other respects, women with AIS look and behave like other women. Some people seem to change sex at puberty. There is a rare genetic mutation that disables the enzyme 5-alpha reductase, which converts testosterone to DHT. These XY individuals develop testes and normal male internal structures, but their external genitalia are not fully masculinized. This is because the genital epithelium is unable to amplify the androgenic signal by converting the testosterone to the more active DHT. The phallus resembles a large clitoris, while the genital folds resemble labia, although they contain testes. There's a village in the Dominican Republic where many families carry the mutation, causing 5-alpha reductase deficiency. When children are born, they look and are treated as girls, but when they reach puberty, their external genitalia become more masculinized and they begin acting like young men. These individuals are called goidosis. How should we define gender? By genes, gonads, genitals, or the brain? Almost all individuals display some behavior that are more common in the opposite sex. Physical features can be confusing criterion to judge sex. Sex, gender, and sexual orientation are distinct and independent from one another. Individuals may be relatively masculine in some traits and feminine in others. Gonadal hormones direct sexual differentiation in the brain and behavior. Testicular hormones direct masculine development of the body. They have similar influence on the brain. Female guinea pigs display lordosis posture in response to male mounting during the time of ovulation when fertility is highest. When males mount at other times, females do not show lordosis. In the experiment, experimenters wanted to induce lordosis by injecting ovarian steroids in a sequence similar to that of which occurs in ovulation. They found that females would display lordosis in response to male mounting. However, female guinea pigs exposed to testosterone in utero and as adults did not show lordosis at all, not even if they were subjected to the treatment that induced lordosis in the other guinea pigs. Researchers inferred that the same testicular steroids that masculinize the genitalia during early development also masculinize the developing brain. The lasting change due to steroid exposure is known as the organizational effect. A steroid only has this effect when present during a specific sensitive period, usually in early development. Early testicular secretion result in masculine behavior in adulthood. The organizational hypothesis gives a single explanation for sexual differentiation, which is that single steroid signal diffuses through all tissues, masculinizing the body, brain, and behavior. If the nervous system doesn't detect androgens, it will organize mostly feminine. Full masculine behavior requires androgens in both development and adulthood. The exogenic metabolites of testosterone masculinize the nervous system and behavior of rodents. After the organizational hypothesis was published, researchers reported paradoxical findings. Newborn females treated with estrogen did not show lower doses, whereas females treated with testosterone in adulthood show mounting behavior. Estradiol is a principal metabolite of testosterone. Aromatization uses enzyme aromatase to convert testosterone to estradiol. The aromatization hypothesis suggests that testicular androgens enter the brain and are converted into estrogen there. These estrogens act on neurons to masculinize the developing rodent nervous system. Brains of females are not masculinized because of a blood protein called alpha fetoprotein, which binds estrogens and prevents them from entering the brain. Testosterone, however, can bypass the alpha fetoprotein and enter the brain. There it is aromatized to estradiol. Several regions of the nervous system differ between males and females. Male and female animals act differently, which would mean that their brains are different. The organizational hypothesis states that this is due to an androgenic masculinization of the brain. Obvious sex differences in the brain include neuronal numbers, sizes, and shapes. Sexual dimorphism describes a condition in which males and females show pronounced sex differences in appearance. Song control regions in male birds. Male canaries and zebra finches sing songs to attract mates, while females do not. The brain nuclei that control song is 5 to 6 times larger in males than females. The robustus archistratum controls motor neurons innervating the syrinx. The RA is controlled by high vocal center. Exposing females to testosterone or estradiol cause the development of large male-like HPC and RA. If they continue to receive testosterone in adulthood, they will sing. However, if the female only receives androgens during adulthood, it will not sing. The preoptic area of rats. Sexual dimorphic nucleus of the preoptic area is more evident in male rats than females. Males castrated at birth had smaller sexual dimorphic nucleus of the preoptic areas in adulthood, while females androgenized at birth had a larger male-like sexual dimorphic nucleus of the preoptic area as adults. This system conformed to both the organizational hypothesis and the aromatization hypothesis. The spinal cord in mammals. The system demonstrates the power of the androgens to mold the brain in sex-typical ways, despite the crucial role of androgens in masculinizing sexually dimorphic nuclei in the nervous system. Experience also affects these systems.